Welcome back to The Explainer, everyone. Today, we're diving straight into Chapter 1 of our text to explore the world of biotechnology. Now, I know when a lot of us hear that word, our minds immediately jump to some crazy sci-fi movie scenario, right? But as we're going to see today, that's not quite the reality. Instead, biotechnology is actually this fascinating, ongoing continuum of human ingenuity that literally spans thousands of years.
Here is our roadmap for this explainer. We're going to start by decoding biotechnology, run through the historical timeline, check out some modern biotech applications, and wrap up with a look at biotech in India.
All right, let's jump right into Section 1, Decoding Biotechnology, from Ancient Roots to Modern Science.
So let's just break down the word itself to start. It's a combination of bio, meaning biological systems or processes, and technology, which refers to the methods, systems, and devices we use. The absolutely crucial takeaway here is that biotechnology simply refers to technologies that use living cells or biological molecules to generate useful products for mankind. It's really that fundamental.
And if you're wondering where the word actually comes from, we have to look back over a century. The term was first coined by a Hungarian scientist named Karl Erecki, all the way back in 1917. This quote here was actually the title of his book, and I think it brilliantly illustrates the roots of the whole discipline. I mean, long before we were doing DNA sequencing, Erecki was already describing how technology could systematically transform plants and animals into useful products for society. You know, it's just really fascinating to contrast those ancient practices with modern science. We've actually been practicing a form of biotechnology since we first domesticated sheep and cattle back in the Paleolithic age. Think about early Egyptian farmers conserving plant stocks, or the classic fermentation techniques we use to make bread, cheese, and wine. That is all ancient biotechnology. Modern biotech, on the other hand, is this deeply multidisciplinary beast. It involves complex knowledge sharing between cell biology, microbiology, computer science, and especially recombinant DNA technology. We've literally gone from baking bread to editing genomes.
Which brings us to Section 2, the historical timeline, the relay race of genetics. This timeline literally plays out like a high-stakes relay race. Just look at how rapidly the baton gets passed. Between the 1850s and 1880s, Louis Pasteur gives us pasteurization, while Gregor Mundell is busy figuring out the principles of heredity with his pea plants. Then, in 1869, Miescher isolates what he calls nuclein from white blood cells, which we now know contains nucleic acids. And then the race just explodes in the 20th century. By 1952, the Hershey and Chase experiment definitively identifies DNA as the genetic material. Literally a year later, in 1953, Watson and Crick proposed the famous DNA double helix. In roughly a century, we sprinted from cross-pollinating peas to unlocking the actual blueprint of life.
Incredible, right?
All right, Section 3, Modern Biotech Applications, from micro to macro. So, what does modern biotechnology actually look like today? Well, thanks to rDNA technology, it exhibits a truly vast range of applications. It's fundamentally changing massive global industries. We're seeing absolute revolutions in pharmaceutical and therapeutic research. It's driving rapid disease diagnostics, crop improvement, the production of vegetable oil and biofuels, and even creating environmentally friendly products like biodegradable plastics. It touches almost every single aspect of modern industry. If we look specifically at crop improvement, and the text handles this very objectively, there are some highly successful scientific applications you've probably heard of. These are genetically modified or GM plants. You've got BT cotton, engineered specifically for pest resistance. There's the flavor saver tomato, designed to have a longer shelf life. And of course, golden rice, which was modified to contain much higher levels of vitamin A precursors. These are classic textbook examples of genetic manipulation applied directly to global agriculture. Now let's zoom way in, down to the microscopic level. Biotech plays a huge role in environmental protection through something called ecotoxicological biomarkers. It's a big word, but basically, these are naturally occurring molecules that tell us how an organism is reacting to environmental or chemical stimuli, like foreign chemical substances. We use these incredibly precise biomarkers to detect very subtle and often completely invisible toxicities in our environment. They are basically nature's warning lights. Let me give you a really cool, concrete example of this from the chapter. Scientists take the lux gene, which is literally the gene responsible for emitting light, and they express it inside E. coli bacteria. This modified E. coli now acts as a living biosensor. So when it comes into contact with specific environmental hazards like mercury contamination, it actually glows. Think about that. We're using a specific gene to literally illuminate toxic hazards in the real world.
Stepping back out to a macro level, there's another incredible real-world application called phytoremediation. We all know heavy metal contamination in soil is a massive health hazard, but nature, aided by modern biotech, actually provides a solution. Phytoremediation uses specific plants, known as hyperaccumulators, to act like giant living sponges. They literally soak up heavy metals right out of the contaminated soil and safely sequester them away inside their own cellular compartments.
And the crazy thing is, we're talking about plants you probably already know. The text specifically highlights examples like Brassica napus and Helianthus annis, better known as rapeseed and the common sunflower. Extensive research is currently identifying the exact genes in these plants that allow them to tolerate and soak up dangerous metals like lead and mercury. By understanding those genes, we can potentially supercharge their ability to clean up our environment. Moving into our final part, Section 4, Biotechnology in India, Academic Prospects and the Industrial Scenario.
To support all of these amazing modern applications, India is actively building a massive institutional framework. Just take a look at the sheer geographical spread of the Department of Biotechnology's research institutes. You've got the Center for DNA Fingerprinting down in Hyderabad. Up north in Punjab, there's the National Agro-Food Biotechnology Institution. Out east in Manipur, the Institute of Bioresources. Literally, from Haryana to Odisha to Maharashtra, there is a dedicated, nationwide infrastructure designed to push the very boundaries of the life sciences.
So, to wrap up our explainer of Chapter 1, the tools of this science have obviously become incredibly complex. We went from ancient cheese fermentation to programming E. coli to glow in the dark around mercury. But the fundamental goal, using biology to improve human life, remains exactly the same. So I want to leave you with this provocative question pulled directly from the text's exercises to keep you thinking. How does the ancient concept of biotechnology truly compare to the modern one? Thanks for watching, and we'll see you in the next chapter.